walking over at this point, using like napkins. See, what's so sad is that I'm such a fast.
So the mic is recording, so it should be okay. And then it just.
Here you go, princess. I can't hold it.
Set this up.
I'm gonna have to interject. I will eat every cookie there.
I wasn't sure where you wanted us to hand this in. This was from last class.
Okay. Thank you.
And she said that she was half last year, and she had a anaphylactic shock, and she brought
Okay. You got my message right? It's the same link that we used last time.
I hope. I'm hoping. Can I just double check on how to? So I just have to turn this on and it's sound, right?
You can access it on YouTube. Yeah, it's public. It's public on YouTube on the channel, so it should be fine. There's just no link.
Okay. Hi everyone. Okay. Yep. Sounds good. Thank you. Thanks for. I'm not okay, but I'm good. No. No. I think if you're around for the second half and you have like other crisis training or something, that would be awesome. Sorry. So I just need to flip it back to the presenter mode. Okay. Thanks. I think we're good. Yeah. Yep.
You might want to make a post on Quarkus just that because I don't think the link is posted.
Nope. So do you want to just post it on the discussions page?
Yeah, sure.
Okay. I'm just not sure why it's not pairing up.
I noticed when I was looking at it, it was only on out of the bed, so the other monitor just died.
It's dead. Okay. All right. Well, we'll just go with it then. Thanks for your help. No, it's a recharge. Yeah. All right. Thanks, guys. All right, everyone. Okay, folks. So if you don't mind, we're gonna get going. I have to be honest, I'm like really nervous today for a lot of different reasons, so it'd be really helpful if we could get going. If I have to pause today, please understand, like I am like really nervous for a variety of reasons, and then if you need to leave at any point in time, like no judgment, like feel free to leave. The first part of the lecture will be recorded and it will be available for you. It's the only part of the lecture that you're responsible for on the term test, so hopefully that will be good for you. If you would like to stay and have a discussion about mental health, please do so, but don't feel obligated to do it. I will be sharing my own sort of discussion about mental health and how it's impacted me here at the university and how it might be important to you as well. So just a quick reminder, at about 4:44 this afternoon on Quirkus, you will have like the previous term test available to you. It'll come up automatically for you, and I would ask you just to use it for study purposes. Not all of the material is going to be relevant to you because we have changed it a little bit from previous years. However, I'm happy for you to go through it. If you cannot write on October the 9th, for whatever reasons, please let the office know. So email hmb.undergrad@utoronto.ca and my email. Let us know by Friday 12 noon that you would like to write it on October the 18th instead. And that's a Friday afternoon. I believe it's either 1 or 2 o'clock. I am not 100% sure. You are welcome to do so. I do not need any kind of any kind of documentation. You're free to do it if things are piling up for you, if you have like other appointments. I'd like you to have that opportunity. I can guarantee you it'll be the same level of difficulty, and some of you just want to get the midterm over with and go on to study other things, so you'll have to decide for yourselves. Okay, so I'm going to take sort of a couple of minutes to go over a couple of things that are really important, and I'm going to try to tie in a lot of what I talked about last week into what we're going to start looking at again this week, and there are some more concepts. I didn't cover this last year, so I need you to pay a little bit more attention, and you need to understand a few things from this lecture. Hopefully, if you've been understanding and following most of what I've been telling you about the gut microbiome, most of it will make sense, or most of it will kind of coalesce for you a little bit. So if you had that moment, that's a good sign that you are in pretty good shape for the final exam. For the term test. So we've been talking about the gut microbiome being a prerequisite, and we know that different types of bacteria in your gut will actually produce different types of short-chain fatty acids. And these short-chain fatty acids, in turn, can somehow regulate microglial expression in the brain, and the changes in microglia and the resulting changes in all of the different types of proteins like cytokines and others that get released from microglia that cause neuroinflammation are thought to underlie the pathology of Parkinson's disease. And that's the hypothesis that we've been working on, and this is sort of a relatively new idea that people have been thinking about. So we went through different types of animal models like the germ-free alpha-synuclein overexpressing mice and the germ-free wild-type mice, and then we also looked at specific pathogen-free mice, etc. The short-chain fatty acids, at least in this paper, were enough to induce neuroimmune modulation and activate microglia in the brain. So keep that in mind for the time being. And it also affected the actual movement, so it exhibited Parkinson's-like changes in its phenotype. We also know that if we look in humans in Parkinson's disease, there are a couple of really striking features of Parkinson's disease in different scenarios. So these Parkinson's disease patients also have altered microbiomes, and in general, when we look at the microbiomes of individuals with Parkinson's disease, we see that there is a reduction in both the number and diversity of bacteria that live within the gut. Okay, so we see that there is indeed a really big alteration. And we know that if we do a transplantation of the fecal matter and the microbiota that comes from a Parkinson's patient and we implant it into an alpha-synuclein overexpressing mouse, that it will also develop Parkinson's-like phenomenon. And here, I had mentioned that all you need to really take away from all of this, one slide that I have up here, is that if we're looking at 16S ribosomal RNA sequencing, and it's not RNA sequencing, by the way, please be really careful. There's a little small r before RNA sequencing. This is not RNA-seq. Some of you will do that in your labs, and that's a different type of sequencing and different type of transcriptomics. This is rRNA sequencing. And when we do that, we can actually plot out the diversity, the number, and the changes in the different groups of bacteria. And what we know is that Parkinson's disease patients, instead of being different, instead of having different enterotypes, which I'll come back to later, so everyone has a unique enterotype, lecture 1. What we find in Parkinson's disease patients, they all kind of have the same microbiota in terms of the 16S ribosomal RNA sequencing. So they look very similar to each other, reduction in numbers, reduction in the diversity of all of the different gut microbiota. And we also know that if we look at how these gut bacteria that have been altered in Parkinson's disease, how it all works, one of the things that we see is that some of the short-chain fatty acids are indeed affected. So we get lower amounts of acetate, and we get higher relative amounts of propionate and butyrate. And these are the short-chain fatty acids that somehow get up to the brain, and somehow in the brain will activate microglia, and after activating these microglia will induce Parkinson's-like changes in movement. So it says that there is a role for all of this. Keep in mind that this study, one of the major points to this is that if we look in our mouse model, and if we look in human models, all of this suggests a couple of things. One, you still have to have the genetic predisposition for Parkinson's. That's what this study is showing. We are overexpressing alpha-synuclein. That is a common variation that we get in Parkinson's. So if you get a gut transplant of Parkinson's microbiota, are you going to get Parkinson's disease? This study is not saying that you'll develop Parkinson's disease. You still need the underlying genetics, okay? So let's be very clear on that. Is that okay for everyone? So the gut microbiome by itself and only by itself in this study is not enough to induce Parkinson's. If you have the genetics, the alpha-synuclein overexpression, and a change in the short-chain fatty acids that result from a change in gut microbiota, then it results in Parkinson's. So again, there's a lot that we've talked about, and I wanted to just spend a few minutes to make sure that we're all on the same page. There will be quite a few questions that come from this section. Do you have any questions before I go on, or anything that really wasn't resolved for you, not clear, things that you just want to confirm, make sure that you're on the right track? Do you have anything that you want to go over? Yes. Okay, so your colleague's question at the front was, so this is like one paper that shows that something changes in the gut microbiome, or the gut microbiota, the bacteria, and as a result there's a change in metabolism, a change in short-chain fatty acid production, and this results in Parkinson's-like phenomenon. Is this the only model, or is this, are there other models? And indeed there are other models available. Do you remember there was this hypothesis that things from the gut travel up to the brain? Do you remember that hypothesis? So it's Brack's hypothesis, and it uses the vagus nerve. This does not use the vagus nerve. It's just producing short-chain fatty acids. There's nothing that starts in the gut and is being transmitted up the vagus, okay? So let me, again, refresh your memory on that. Yes, Lauren. Is there a process? So here in this study, what they're suggesting is that there isn't any misfolding that's occurring in the gut, and this misfolded protein isn't traveling up through the vagus into the brain. Other people have hypothesized that, and that would be Brack's hypothesis, that if you cut the vagus and it started in your gut, and there's alpha-synuclein in your gut, and that it's misfolding in your gut first and then traveling up the vagus into your brain and then causing other misfolded alpha-synuclein in your brain. There is that hypothesis too, but that's not what they're showing here. Yes? Okay, so what causes the changes in the different short-chain fatty acids? And I'm going to try to address that in a few slides. Do you have any other questions? Yes? Sorry, you're going to have to speak up just a little.
How physical is this model, knowing that only a few patients?
Right. So how useful and how applicable is this model, knowing that most of the cases of Parkinson's disease we just don't understand. We don't have the genetic sort of underlying cause. We only know of a very few, Parkin being one, alpha-synuclein being another. We don't really have a lot in terms of genetics. So how does knowing all of this, that somehow the gut microbiome changes the short-chain fatty acids that result in Parkinson's affect like the general population? We don't know, but if we were able to detect individuals that have a genetic predisposition, then it might be a benefit to those individuals, whether to get a fecal matter transplant or to receive different types of short-chain fatty acids. Okay, so those were excellent questions, and thank you for these. The next slide that you have in your slide deck is a little bit different because I've added some more material, and I'm going to ask you to write that down. I'm going to spend a few minutes going over this, so if you have any questions along the way, please let me know. Most of you will have heard of autism spectrum disorders, and these autism spectrum disorders are thought to be a neurodevelopmental condition. Something goes wrong, resulting in a lack of social awareness and uncomfortability with like social contact and communication, along with a number of other things that are very characteristic of autism spectrum disease. Have you all heard of ASD or autism spectrum disorders? Yep. Okay, and if you don't, I'm going to try to go through some of these really slowly later on. These are individuals that really have difficulties in different environments that need social interactions. They really can't communicate well, and so it makes it very awkward and very challenging for them to learn or for them to interact with others. And it's thought that it's irreversible. But here's this interesting paper that came out not too long ago, about a year and a half, 2 years ago, that actually suggested that in autism, similar to what we see in Parkinson's, we can actually reverse some of the different types of communication and other issues that are found in autism. To me, that was really surprising because once you have a neurodevelopmental condition, and once everything is hardwired into place, that all the axons have found the appropriate connections in the cortex, etc., and your circuits are formed, that you can actually modulate that circuit again after you're an adult. Or when you're older and that critical period has been elapsed. And so here this particular paper had a lot of really interesting and very surprising things for us to kind of keep in mind. This is also sort of the next generation of what we are now looking at in terms of the gut microbiome. So if you take a look at this paper, and I would say that it would be an excellent paper for you to do a review on if you're interested in this. One of the things that we now do that's different than it was 5 years ago or 6 years ago when people were starting to think about the gut microbiome if you are going to get a gut microbiome transplant, so you replace the bacteria here, there are a couple of things that are now done, that are done in this paper that actually help. And this is something I want you to be aware of. So one of the different types of antibiotics that are used is vancomycin. So it reduces some of the more toxic pathogenic bacteria that some individuals with autism spectrum disorder seem to have a little bit more of. So it kills those off specifically and selectively. So it's sort of important to include vancomycin before you do the gut microbiome transplant. In addition, you also will add a substance known as movie prep. And this is sort of to clean out any residual bacteria that's left that might be toxic in the environment that you're transplanting back into. We didn't used to do that, and now it's sort of more routinely done. We're learning a lot about how the gut microbiome works and how to get good colony formation. So previous studies where they didn't see an effect, they weren't doing these types of things, and now these types of things are being added. Do any of you know the drug Prilosec? What is Prilosec? Without looking it up, without talking to your friend Google or Siri. Any ideas? So let me give you a little bit of a clue. So this is taken orally. So this is given by gavage. So there's a tube implanted and it goes through the stomach. Does your stomach like bacteria? Your stomach likes bacteria in general? So I don't know who teaches PSL 301. Who teaches the gastrointestinal system in PSL 301? Dr. Wong. So Dr. Wong would say no. No. So your stomach is designed to actually resist bacterial colonization, except for H. pylori, which loves acid. But your stomach is acidic, right? And the acid actually helps to combat any bacteria in the food that you're eating from colonizing your stomach. And so to prevent that, when you're introducing new bacteria, what we do now is to also administer Prilosec. And Prilosec is used to reduce the acidity and the acid production in the stomach. So all of these things enhance the long-term colonization of this new microbiome, the new microbiota that's being delivered. So let's go through some of the different aspects of this paper, which may be interesting to you or may not be interesting to you. And along the way, let's go back and revisit some of the things that we saw here earlier. Hopefully it'll jog your memory and it'll also give you some insight into what you need to know for the upcoming term test. Do you remember how many times, if we believe the current model system, how many times has your gut microbiome changed as long as you were a healthy individual from the time of birth? 3. So here you're looking at from the time of birth on the left, and then we're seeing that in early infant stages and then toward the time that you're eating solid foods, those are the 3 different stages that you're seeing. And as long as you're healthy, what you have now, there's a balance between the bacteroidetes and the firmicutes that have accumulated during the 2 to 3 years of your life. So this is what we're looking at in this particular graph. And one of the things that we know is that in autism spectrum disorders, there's something wrong with the gut colonization. They also tend to have comorbidity with irritability of their gut, or they have issues with their gut, or they have like gastrointestinal symptoms, which kind of make it a very interesting sort of segue into maybe using gut microbiota to change some of those symptoms. So some of you were asking, like, where do these short-chain fatty acids come from, and what is the purpose of these short-chain fatty acids? So we'll play the usual game. I'm a little bit disoriented because you guys are sitting in the wrong place today. So do you need to know this slide for your upcoming term test? No. That was like the worst no I've ever heard. No. You do. So I'm going to tell you ahead of time, you do need to know this slide. I'm going to tell you like what you need to know. So going back to Dr. Wong, PSL 301, when you start the process of starch digestion and you break it down into smaller polysaccharides, then into oligosaccharides, eventually what's left by the time it gets to the the large intestine in particular, you're left with a lot of these monosaccharides, right? So these monosaccharides, for example, if you have bacteroidetes and lactobacilli, or if you have bifidobacteria in your large intestine, this will lead to the production from monosaccharides into lactate and into acetate. So all of those different bacteria that we talked about, like bifidobacteria, and the production of these different breakdown products like lactate and acetate are kind of important. And the other thing that we talked about is that there's a balance between these bacteroidetes and these firmicutes. In other words, there's a balance between having too much of these bacteroidetes as well as these firmicutes. And why that's important is that some of these different firmicutes that you see here will be able to convert some of that lactate that's left over, or the acetate that is a byproduct of monosaccharide breakdown, into propionate. And there's one of those short-chain fatty acids that we have just been talking about. Some of the firmicutes may, depending on the type, can also convert them into butyrate. And I don't need you to know the difference between Clostridium types that are isolated here. I just want you to make sure that you know that this balance between bacteroidetes and these firmicutes can result in changes to the breakdown products like propionate as well as butyrate. And the last one that we're going to consider is that if we're looking at some of these other breakdown products, and I mentioned this in passing, some of these proteobacteria are actually sulfate reducers. We don't really concentrate too much on the right-hand side. I'd rather you focus on the left, just for your own information, that some of the different sulfate reducers are also ones that we've covered before, like proteobacteria. But for the term test, I would suggest that knowing this part of this pathway is the more important one, because these are the short-chain fatty acids that we've looked at in this course. And you should know the types of bacteria that we are looking at in these breakdown products. So let's take a look at some of the data that actually comes out of this paper. And you don't have to memorize this data. In fact, I'd rather you kind of just concentrated on what this data is showing. I won't be giving you a graph and asking, what does this show? I just want you to see the positive correlation. So on the x-axis in every single case that we are looking at here, we are looking at the percentage change in these gastrointestinal symptoms on a particular rating scale. So I told you earlier that many individuals that have autism spectrum disorders have something wrong. Their gut just doesn't feel very comfortable, or they have too much bowel movements, or they could have like constipation, or they feel bloated. And those are different examples of gastrointestinal symptoms. And these often accompany some of the things that people with autism or autism spectrum disorders have. And after 2 years after a gut transplantation, they're seeing a more positive outcome. So they don't have the symptoms of having like gastrointestinal upset. So that gets fixed. In addition to that getting fixed, we also see a change in different measures, like the childhood autism rating scale. They perform better after time, after getting a transplant. Now, the interesting thing about this study, which is, again, for me, I was looking at it and I was going, wow, you know, the results are amazing if these results can be replicated. They have not been replicated yet. So they did this study in humans. So that's the first thing. It's not an animal model. This is all human data. And what they were looking at is a very small window where they were introducing a normal gut microbiome, not the one that they have currently where they have autism. They replaced their gut microbiome using vancomycin, movie prep, and then they also blocked acid production. And then they did that for 2 weeks. That's it, 2 weeks. And then they saw improvement. But more surprising, this study follows up 2 years later. So they stopped after 2 weeks. And then they followed up on these patients like 2 years later. So the gut microbiome colonized. It actually took root, and it actually produced these long-lasting changes that are occurring like 2 years later. For me, that's a remarkable feat. No one has been able to do that, as far as I know, in these types of papers. And in every instance, whether we're looking at childhood autism rating scales, where we see that they're performing better. So as their stomach gets better, it's a positively correlated outcome. They're also getting better sort of childhood autism rating scales. They're also much more socially responsive, so they get more social interactions. And this, again, is remarkable. This is 2 years later. They're still seeing a positive effect. They're getting better. And all it took was a 2-week window where their gut microbiome was replaced. In addition to that, and this is a busy slide, and you don't need to worry about all of the details on this slide, and apologies, I wanted to just show the raw data because it is very messy raw data. And here, if we're looking, and here's an opportunity for me to ask you some questions. What we're looking at here on the y-axis is what is known as phylogenetic diversity. So one of the things that we're always interested in, is it better to have more diversity or less diversity? So generally speaking, we want more diversity in the gut microbiome. And what we see with this transplantation, 2 years later, they've actually increased the overall diversity versus what they began with. So we see an increase in diversity, and there are different names depending on the different investigators that are using it. This is known as Faith's Phylogenetic Diversity Index. You'll see other diversity indices. How did they get this, a measure of this diversity? What kind of technique did they use? We've already covered it once today. So this 16S ribosomal RNA sequencing allowed them to quickly screen all of the different types of bacteria, and they're seeing that they get an increase in diversity 2 years after the fact. So they started improving, and this is still there 2 years later. And again, pretty remarkable if you think about the long-term benefits, and their autism features are also getting better as well. So a couple of interesting notes, because we just talked about bifidobacteria, and we've also previously talked about Prevotella in an earlier lecture. So after they had done this gut microbiota transplant, they actually saw that there was an increase in bifidobacteria and an increase in Prevotella in the gut microbiota of individuals after the transplantation. Another species, the sulfobribrio, also decreased. So it suggests that these might be important. They didn't follow up on this. This was more of a report as to what might happen in these gut microbiomes, but it was pretty remarkable. I don't know if you are like stunned by that. Like for me, when I read that paper and seeing these long-lasting changes, a fairly easy therapeutic intervention, it gave me a lot of hope that maybe some of these different types of disorders can actually be reversed. We didn't think so 5 or 6 years ago, but this data is pretty convincing. Did you have any questions on this before I go on? No? Oh, okay. So I'm going to go back and kind of fill in a couple of other missing pieces. Again, I have to apologize in advance. You are going to get this material on your upcoming term test. I can tell you in advance that one of your multiple choice test questions will involve you knowing different parts of what I'm going to tell you. One of your short, like one-line answer questions also will be from here, and then part of your short answer will also come from here as well. So I want to make sure that you understand what I'm going to be explaining to you. So a few years ago, I think about 1.5 years or 2 years ago, there was this paper that came out that actually tried to use molecular biological techniques to actually trace whether or not there is a connection between the gut and the brain. Because people hypothesize it, and this is Brack's hypothesis, but it's not a really definitive proof. And at least in this model system that I'm going to show you, they were able to show definitively that there is indeed a connectivity between the gut as well as directly into the brain. And there's some molecular techniques along the way that I need you to be familiar with. This, by the way, does not cover it all. Like if you get alpha-synuclein in your gut and then it misfolds in your gut, that it travels up this pathway, but it suggests that this pathway could be involved. There's no evidence here of that misfolding. All right, so let's just dive right in. So we're looking at this intestinal villus. So this is the intestinal villus. Here's another one right beside it. And these little green blobs are stains that have been done for a particular type of cell, which is called a neuroepithelial cell. And these neuroepithelial cells, if you had learned about it back in PSL 301, their primary function wasn't really well described. And we didn't really know, other than the fact that they might be releasing these enteroendocrine molecules like CCK, cholecystokinin, or PYY, that outside of storing and releasing these, and that might have sort of a local effect on the stomach, like what else they were doing. But people suspected that they actually had another function in addition to this enteroendocrine function. So here again, in the intestines, we have these little green labeled cells, 1, 2, and 3. And these, again, are cells that are neuroepithelial cells. Previously, they were identified as being enteroendocrine because they were thought to release and only release CCK or PYY. But then investigators started to use some molecular techniques to suggest that maybe this is the route by which a signal from your gut could actually transmit information through the vagus nerve, because the vagus nerve also innervates this. It makes a connection onto the cell bodies of the vagus nerve ganglia at the nodose ganglion, and then it will directly enter into the brain. So here's this roadmap. And they were able to show that this is a direct connection between the vagus nerve. It confirms Brack's hypothesis that your gut is indeed, there's a physical connection to the brain, and this is the connection that they were able to show. So how did they actually demonstrate that this occurs? Hoping this goes. Sorry, let me just hope that it's going to go. Okay, one thing before I go on, if you were to go back in and examine this neuroepithelial cell, also called a neuropod cell, so these are interchangeable terms. In PSL 301, you would have called it an enteroendocrine cell, and you would have thought it released only this. In fact, if you look at the ultrastructure, you look at it in more detail, you will actually be able to see presynaptic proteins. In fact, you will see synapsin 1 staining at the level of this neuropod, neuroepithelial, enteroendocrine, whatever you're calling it, type of cell, and it almost looks like it's making a synapse, which made scientists suspect that maybe it's doing something more. So one of the things that we need to show is that this actually makes a direct connection. Sorry, something is going on with the computer, so let's just give it a second. Okay. So most of you will have learned that epithelial cells and neurons are particularly vulnerable to being infected by viruses, right? So you can get a viral lesion on your skin, or you can get a virus traveling up a nerve. And one of these types of viruses is the rabies virus. And there's a genetically engineered rabies virus that was developed, and this rabies virus also contained GFP. So if I have the rabies virus, and this rabies virus also contains the sequence for GFP, and it infects a cell, what color will that cell fluoresce? Green. Okay. And this delta G rabies virus actually is a special variant of the rabies virus. So if you know anything about rabies, if you get bitten by an animal that is infected with rabies and it has rabies virus in the saliva then enters in to your skin. From your skin, it can then travel along a nerve and then into your own central nervous system and infect your own central nervous system. And that's the way these viruses work. They genetically engineered a special version of this rabies virus called the delta G rabies virus that also included GFP. And the reason why they did this was, first, they wanted to confirm that it could indeed infect a neuroendocrine cell or a neuroepithelial cell, and they were able to show that it was selective. So out of all of the different types of intestinal cells, all of those different cells that you see here, we only get a few that will take up this rabies virus. So not everything is green, only the ones that are neuroepithelial will take up this green virus. Is that okay for everyone? Are we all on the same page? Are we not on the same page? Same page. Different page? No. Okay. Same page. All right. It really doesn't like this one slide. Okay. So it showed that it's there. It infects it. It infects it very, very specifically, and that this thing does not spread. It's trapped in that cell. And the reason why it can't spread, like the normal rabies virus can spread, is that it is missing a key feature of the packaging of the virus. So it can enter the cell no problem, and it makes the cell get infected, but it can't actually spread to the next cell that it would normally connect with. And that's why it has this delta G associated with it. It is missing what is known as the G glycoprotein. Without this G glycoprotein, a key element in rabies virus packaging, it can't actually jump from cell to cell. So it's been trapped in that particular cell. No big issue, because here in A, all we want to do is show that the infectivity is very, very specific to that group of cells. Not everything is going to be infected, only the neuroendocrine or neuroepithelial cells will be. So then they go on and show after that experiment that indeed, if you supply this cell with the synthetic pathway that will allow for G glycoprotein to be produced, and this is RABG, and they genetically engineered a group of mice that have RABG in these cells. If you introduce this delta G rabies virus, and these cells, only the neuroepithelial cells are expressing the synthetic pathway RABG that will produce G glycoprotein, this cell gets infected again, as we'd expect. In addition to that, it is now fully packaged because the RABG allows the G glycoprotein to be incorporated into the rabies virus. So this delta G is no longer an issue. We have the RABG filling in for it, and this virus can now spread to the next cell. So whatever cell is connected to this, we're now able to trace. How many of you are lost? Oh, wow, that was a very confident hand raising. Okay. So let me go back. Was this okay? Kind of okay. So do you know that viruses can infect cells? Rabies viruses can infect epithelial cells and neuronal cells. And here we have a neuroepithelial cell. And we have genetically modified the rabies virus so that it infects a cell, but it can't spread beyond that one cell that it infects. And so using the delta G rabies virus, we're able to show that, yeah, there's a group of cells that are neuroepithelial that lie in the gut when we introduce it through like gavage. Okay, so we're not injecting it, we're just introducing it into the lumen of the gut. So are we all on the same page here? Okay, that was also not a, is it this or is it thumbs up? Thumbs up, okay. So now we want to know, what is this cell actually connected to? The rationale for this, why we want to know that, is in the previous slide, which I'm not going back to because my computer is freezing up, we have these structures that kind of look like synapses. We have synapsin 1. We have this one cell that we were able to infect. It's fluorescing green, and it kind of has this structure that looks like a synapse. And it has synapsin 1. And we want to know, what is that cell connected to? And one way that we could see that if this cell is infected, and we have a virus that can then infect the other cells that it's connected to, we might be able to show and demonstrate that. Does that rationale make sense to everyone? It doesn't make sense to everyone. Okay. Repeat the last sentence. I can't remember the last sentence. Okay. So the delta with the delta rabies GFP, the cell itself gets infected, but It cannot infect any other cells. It's trapped in that one cell. That cell, we get very, very few of these cells being infected, and these cells are neuropods or neuroepithelial cells. And these neuroepithelial cells, very small numbers of them, so remember that intestinal villus in blue, there are literally millions of other cells, but we only get 3 of them actually lighting up. Those cells in green actually allow for, or have a structure that looks like a synapse, and it contains synapsin 1. So might that be of interest to us? It might be. Sorry, I see your hand up at the back. You're going to have to speak up really loudly because I'm not so confident with the microphone system. Or which table number are you? G11. Sounds like E11. Okay. Wow, so many different things. Okay, let's try that. Okay, I think your microphone's now active. Yep, I can hear you.
So I was just wondering, what's the point of tracking the viruses at one cell, only for you to reintroduce the reaction protein later and for you to trace it? Because the purpose of it is to trace the. So what's the point of tracking it here?
So why not just jump right into like a normal full rabies virus and then see what else it's connected to? Okay. Okay. So one of the reasons why we want to make sure about its specificity is we want to actually see that it only infects these cells first. And if we just started from the beginning, we wouldn't know if it infected the other cells in addition to this one. So if I see like green everywhere right from the beginning, I wouldn't know if it was infecting A and B together, or if it was A and then it traveled to B, or if it was B and then it traveled back to A. Here, I can temporally determine what cell was infected first and which direction this is going. Does that make sense to you? Does that make sense to everyone? Okay. So allowing for the full expression and packaging to make it more infectious, we will have a genetically engineered neuroepithelial cell that expresses RABG, and this RABG will provide the missing G glycoprotein so that when we now go in and do the same experiment with this delta G rabies GFP, which is normally lacking this G glycoprotein, and without this, it is remained trapped inside this one cell. By having this expressed, this RABG that will form G glycoprotein in this cell, it allows for the virus itself to be fully packaged and then fully infectious. And so it will spread to the next cell along its pathway, and it will spread to the next cell. And this next cell, it turns out, is the vagus nerve. And this was the way that we mapped out the vagus nerve directly innervating discrete numbers of cells within the gut. Now, there's a lot there. Are you guys all good with that? Yes. Yep. 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 So this is a different mouse that has genetically engineered RABG expressed only in neuroepithelial cells. I didn't want to talk about Krelok systems and specific expression.
So the first part has happened in one mouse, the first ones.
Right.
Do we have to do a second?
So here we're looking at a different mouse. Right. And it turns out that when we look, it actually does indeed function as a synapse. This neuroepithelial cell that we thought was only releasing cholecystokinin and PYY is also capable of releasing glutamate directly through those synapsin 1 structures and is able to activate the vagus nerve along this pathway. And so it opens up this new avenue that suggests that Brack's hypothesis that we began with for Parkinson's disease is potentially correct. We still have not in this model looked at how in the gut alpha-synuclein can misfold and then travel along the gut, but it does show that it can happen and that there's a mechanism by which all of this can happen, and this is the way that we would trace that direct connection. Okay, so this is the paper that I'm going to be working on for my own visual summary, it's one of those things that's important because it also introduces the microglia. And this paper is important because it suggests that there's a link between microglia and the spread of inflammation throughout the brain. Once you activate microglia, it's pretty much game over because it spreads through astrocytes and then affects neurons. And this is actually a really interesting and very important paper. So I'm just going to go on for 2 more minutes and kind of wrap things up here in the next 2 slides. So as we sort of transition into the talk that's optional, I hope some of you will stay, but there's no onus on you staying. As we started this discussion early on, the gut microbiome is one that seems to be very important in mood disorders. So we're talking about anxiety, generalized anxiety, maybe even social anxiety, depression, bipolar disorder. All of these different mood disorders have something in common with the gut. And it turns out that if you were, just like we've been looking at autism, we've been looking at Parkinson's, and if we look at individuals that have major depression, we can also see changes in the gut microbiome in these individuals as well. And again, you don't need to memorize everything on this slide. I want to use this slide more to help synthesize some of these different concepts and then give you that little aha moment, hopefully, when we talk about this. Do you remember earlier on we were looking at the enterotype? And remember, the enterotype consisted of a ratio and a difference in the ratio of these 3 different types of bacteria. So the bacterioids, and the Prevotella, and then the Ruminococcus. Do you remember that slide? Well, here is the functional way that we look at it. You all have an enterotype that has different ratios of this. And it turns out that if we look at individuals that have major depression or depression on the right, their enterotypes look different. There's a difference in those ratios, that fingerprint of these different bacteria. And again, using 16S ribosomal RNA sequencing, they were able to show some really striking things. That for you to have like a good quality of life, for you to be happy, for you to feel like you're enjoying your life, you actually need to have the expression of certain types of bacteria, particularly butyrate-producing bacteria, like the Faecalibacterium and the Coprococcus bacteria. These were actually always associated. If you have high levels of these, you generally feel like you have a higher quality of life using the 16S. Individuals that have depression have a very different enterotype, so you can see the changes in all of these ratios that we see here. And in particular, if they looked, they saw that these other bacteria were affected where certain species of bacteria in depression were completely depleted. It's not that they were reduced, they were gone, like they're absent. And so they didn't do anything further in this study. They didn't go back and replace these bacteria to see if they could reverse some of the symptoms of depression, but it was a fascinating study to show that yes, in every disorder that we look at, it may just be it may be correlational, it may not be the cause, but we do see disruptions in almost anything that affects the brain, whether it's neurodevelopment like autism, or whether it is a mood disorder like major depression, or whether it is a neurodegenerative condition like Parkinson's or others. We always see something with the gut microbiome. So I think at this point, because the rest of it is actually more related to the talk on depression, I'm just going to go through this really quickly. So we're going to take a break a little bit late, 2 minutes. And I wanted you to know this slide. So if you know that you need to see this slide, do you think it will be on your term test? You sound very enthusiastic. Okay, yeah, you probably will get a question related to this. So here's this really interesting phenomenon. And again, it's controversial. So some of you who might be taking Psych 307, I think, with Dr. Downer, I don't know if he's still teaching the course. So again, it's a little controversial, but I'm going to give you my take on this. depression used to be thought to be purely psychological. Like, it has nothing to do with the brain. It has something to do with like your psychology. Nothing structurally is changing. But in fact, if we were to take a look, there are significant changes to the brain. There's an impact on the brain of having depression. The longer you have depression, by the way, each of these different symptoms gets worse. So you have less gray matter overall. There's a reduction in gray matter. There's also a reduction in the white matter tracts, the axons, the myelinated axons. And there's also less glia, and we particularly see this within the hippocampus and prefrontal cortex. Why is that important, by the way? Any idea? Yes.
A lot of depression is associated with memory loss.
Right. Thanks, Lauren. So a lot of you will be thinking, depression on the bigger scale of things is actually not such a terrible disorder, but the biggest chief complaint of anyone, especially students, and this is why it's important to you as students. Yes, there's a mood element to it. So yes, you might have things where you're in a down state and you're in a down mood for a very long period of time, like 16, 18 months, 2 years, 3 years. But what impacts students more than that change in mood is actually a loss in cognitive skills and memory. That's the thing that actually impacts everyone more than the mood part of it. That's important, but the loss in memory, the loss in cognitive functioning, decision making, that's actually much more important for these individuals. And this is why the hippocampus with memory is really important. The problem is, most of you are not walking around with a snapshot of your hippocampus when you're in like a really healthy state. Like if any of you have that in your wallets right now, bring them out. I'd love to see them during the break, but most of you don't, right? So you have snapshots of yourself when you were younger, maybe on your Facebook page, but you don't really have a snapshot of your brain. And you should. You should all have a snapshot of your brain when it's nice and healthy. The anterior cingulate also is going to have higher activity overall in depressed individuals. The amygdala is hyperactive, and it also grows in size in individuals with depression, and this is often associated with anxiety. And then the left prefrontal cortex also seems to shrink a little bit and decrease in activity. We don't see it in all individuals, and in fact, there's a discussion about if you see some of these and not all of these, what type of depression is it? But these are real changes that occur within the brain, but some of them are so small that you wouldn't be able to tell that something is awry or something is amiss unless you already had a snapshot of your brain to begin with. And in the next lecture, which you're not responsible for, I will talk about the hypothalamic pituitary axis and how in some individuals this gets affected as well. So that's it for the lecture that you're responsible for up until the term test. There is a review this Friday, if you can make it between like 1 to 2:30ish. And if you happen to be able to come in, that's great. If you can't, it will be streamed and it will also be recorded for you as well. So let's go ahead and take a break. If you are going to stick around, we'll get going in about 10 minutes or so. And then in the interim, why not take the time to get up and meet some of your classmates?